Hello friends, in this video I'll be talking about the mechanism of folate trap. Okay, this folic acid or folate, it is water soluble vitamins, and the major function of this water soluble vitamin is hemopoiesis. That means it helps in formation of the blood cells. It is also involved in DNA synthesis. Okay, since it is a hematopoietic vitamin. Okay, so I'll be talking about how the folate get trapped in the form of 5-methyl tetrahydrofolate. Okay, so during the course of methionine metabolism, this methionine is first converted into S-adenosyl methionine by the enzyme methionine adenosyl transferase enzyme. Okay, and this compound, which is called as S-adenosyl methionine or SAM, is an important compound because during the methyl transferase reaction or the enzyme catalyzing the methylation process uses SAM as a coenzyme. Okay, so wherever there is a methyl transferase enzyme catalytic activity, this SAM serve as a donor of this methyl group. Okay, for example, this DNA methyl transferase enzyme that is involved in the epigenetic modifications of the DNA or methylation of the DNA uses SAM as a coenzyme. Similarly, we have guanidinoacetate methyl transferase enzyme, which is involved in creatine synthesis, also uses SAM as a coenzyme during their catalytic reaction. Okay, so it is mainly it is the main component that is utilized by the methyl transferase enzyme. Okay, so during the methyl transferase reaction, okay, this S-adenosyl methionine is converted into S-adenosyl homocysteine. Okay, and this S-adenosyl homocysteine is converted into homocysteine by adenosyl homocysteinase enzyme. Okay, now the homocysteine that is formed can be reconverted back to methionine, or it can be converted into cysteine by transsulfuration pathway. So this homocysteine is converted into cysteine by transsulfuration pathway catalyzed by the enzyme cystathione beta synthase and cystathione gamma lyase. Okay, and 50% of the cysteine that is formed from this homocysteine is utilized for the synthesis of glutathione, and this glutathione is a part of antioxidant defense system. This glutathione is a part of antioxidant defense system. Okay, so now after the homocysteine it is formed, it can be converted, as I told you, as I said, it is converted into cysteine, or either this homocysteine can be again converted back to methionine. Okay, so this homocysteine to methionine conversion is catalyzed by the enzyme methionine synthase. Okay, and this methionine synthase uses methyl cobalamin, which is a active form of vitamin B12, as a coenzyme. Okay, so during this reaction, this methyl cobalamin donates its methyl group to the homocysteine and it gets converted into methionine. Okay, but after this methyl cobalamin donates its methyl group to the homocysteine, it is converted into cobalamin. Okay, now this cobalamin is a useless compound in the body. So this compound again needs to get converted into methyl cobalamin. Okay, and that here 5-methyl tetrahydrofolate serve as a methyl donor that converts this cobalamin into methyl cobalamin. After donating its methyl group to the cobalamin and it gets converted into methyl cobalamin, it gets converted into the 5-methyl tetrahydrofolate gets converted into tetrahydrofolate. Now, in case of deficiency of methylcobalamin or vitamin B12 whatever the homocysteine is formed during the methionine metabolism can be converted into methionine, because there is no methylcobalamin for that required by the methionine synthase during the conversion of homocysteine to methionine. Okay, and the 5-methyl tetrahydrofolate that is formed during the folate metabolism can get converted into the further different forms of tetrahydrofolate required for the different biological activity, and all those tetrahydrofolate they remains in the form of 5-methyl tetrahydrofolate, and this mechanism or this phenomena is called as the folate trap.